Another situation is called pleiotropism. Pleiotropism. Pleiotropism is a situation where more than one trait is influenced by a given gene. Normally, a particular gene expresses a particular trait or a particular character. We have seen there is a gene responsible for length of the stem. There is a gene responsible for position of the flower and so on. But this is a situation where a given gene not only expresses a particular trait but also has a role in the expression of many other traits. So such genes are called pleiotropic genes. Pleiotropic genes. For example, in the sweet pea plant, Lathyrus odoratus, the gene which influences color of the flower also influences color of the pods. And it also is known to influence the nature of the axil of the flower. It is also known to influence the nature of the axil of the leaf. In some leaves, at the axil, a red spot may be present. The occurrence of that spot is influenced by the same gene which influences color of the pods, which also influences color of the flower. Another example is Drosophila. Drosophila melanogaster is an insect that is commonly called fruit fly. In this insect, there is a gene which decides the wing size. The same gene also influences the lifespan of the insects. So it not only has an influence on wing size, but also has an influence on the lifespan of the insect. There is an example from human beings. In humans, there is a genetic disorder called sickle cell anemia. Sickle cell anemia. Sickle cell anemia is a genetic disorder. In this genetic disorder, hemoglobin will be defective. You know that hemoglobin is a pigment that carries oxygen. Here, due to a mutation, hemoglobin becomes defective. Normal hemoglobin is represented as hemoglobin A. Defective hemoglobin is represented as HbS, hemoglobin S. Not only the hemoglobin becomes defective here, the RBC become sickle shaped. RBC becomes sickle shaped. So due to defective hemoglobin, oxygen supply to the tissues decreases. The RBC has become sickle shaped, and therefore it is called sickle cell anemia. Sickle cell anemia. There is a marriage between a normal individual and affected individual affected with sickle cell anemia. We will represent this as HbA and this as HbS. So assuming that this individual is homozygous for this condition, and this individual obviously is heterozygous for this condition. So the gametes will be HbA here, HbS here. So their offspring in the F1 generation will be carrying the genotype HbA, HbS. It means these individuals will be having the dominant genotype for A, recessive for S. So they will not show the sickle cell condition normal, but they will be different from these homozygous normal individuals. They are carrying the gene responsible for sickle cell condition, so described as carrier individuals. Now suppose this is a case of human beings. So suppose a carrier individual marries another carrier individual. So two types of gametes here, HbA, HbS, HbA, HbS, 2 into 2, HbA, HbS, HbA, HbS, this into this, HbA, HbA, this into this, HbA, HbS, this into this, HbA, HbS, this into this, HBS, HBS. So look at the situation. Normal individuals, carrier, affected. It means in the F2 generation, you will have normal individuals, carrier individuals, affected individuals in the ratio 1:2:1. So the same gene is expressing a different phenotype.